Hi, I'm Dr. Stefanie Chen, and I'm a biochemist, and I study DNA repair mechanisms. Hi, I'm Dr. Stefanie Chen, and I'm a biochemist, and I study DNA repair mechanisms. The backbone of DNA actually breaks regularly just from normal environmental stresses. So when a cell's growing, just the oxidative stress from inside the cell and metabolism, or light, radiation coming in, any of those things can provide enough energy to actually break the backbone of DNA. And if you get a double-strand break, that's really bad for the cell because the polymerase can't replicate the whole genome then. So you really have to fix double-strand breaks or that cell will actually die. There are a number of DNA damage pathways depending on the type of damage. For example, UV light can create some lesions, and there's an enzyme photolyase that will directly repair that. For DNA double-strand breaks, we have homologous recombination, which is where you basically copy the same section of sequence from the partner chromosome. So the enzyme will bind the broken part, and it'll find the similar sequence, and the polymerase will be able to copy that off, and then the two ends can come back together. The result of the homologous recombination repair pathway is that your two chromosomes are actually physically hooked together by these structures called Holliday Junctions, which are these four-way DNA junctions. And there's two ways you can separate those, and you need to separate those chromosomes to get cell division in viable daughter cells. And there's two ways to do that. One, you can just cut them, but there's a chance that a section of this chromosome will be attached to this chromosome, and that's called crossing over. And that increases the frequency of mutations, and those can actually cause cancer if you have too many. So we try to avoid that. And the way the cells do that is they have a mechanism that actually pushes the junctions back together, and those chromosomes largely remain intact except for that repair region. The migration of those junctions to avoid mutations requires two enzymes. One is Bloom's helicase, which is able to migrate the junction, and the other is topoisomerase 3 alpha, or topo3 alpha, and that's an enzyme that unlinks the DNA. So if you were just to push the junctions together with a helicase, you'll actually build up supercoiling in that DNA, and the energy of the supercoiling will prevent the junctions from actually reaching each other. So you need the topoisomerase to relieve that supercoiling to actually push them together. In graduate school, I studied the interaction between the two enzymes, the Bloom's helicase and topo3 alpha, by making mutations in a particular C-terminal region of topo3 alpha to show which regions were important for that interaction. And if you don't have that tight interaction, if you disrupt it, you won't actually get the resolution of the Holliday junctions where they actually come together. As I mentioned, because of that supercoiling, they'll never actually touch. And so that mechanism is broken, and they have to use the cutting mechanism instead. In my current work, I study a gene that we found makes E. coli resistant to radiation damage, but the pathway isn't well understood. We don't know what it's doing. So that's really exciting, but also kind of scary. We have to find out what it's doing in the cell. And it has lots of implications to human health, cancer research, space travel, anything where you're going to get a lot of radiation damage and you want to protect against that.